Welcome to the Biotech Startups podcast by Exceeder. Join us as we speak with first-time founders, serial entrepreneurs, and experienced investors about the challenges and triumphs of running a biotech startup, from pre-seed to IPO, with your host, Jon Chee.
My guest today is eMalick Njie, CEO and founder of Ecotone AI, a company with a vision of AI-designed medicines to cure rare inherited diseases. eMalick is also co-founder of Genetic Leap, formerly known as Genetic Intelligence, a company that is innovating at the cutting edge of AI and RNA genetic medicine to redefine drug development and more quickly address the health needs of millions of people. Before his transition into entrepreneurship and industry, he was a senior scientist at Columbia University, where he discovered multiple C. elegans genes related to neural achievement and sensation of touch in the laboratory of Nobel Prize winning Marty Shelfie. eMalick's diverse experience and deep expertise in neuroscience, AI, and biotech entrepreneurship makes his conversation one that founders won't want to miss. Over the next 4 episodes, we cover a wide range of topics, including eMalick's move from West Africa to the United States, his time at MIT, his experience moving to Florida where he completed his Ph.D. work, the sacrifices he made while founding Genetic Intelligence and Ecotone, and his vision of how to leverage AI to tackle disease. Today, we'll chat with eMalick about his journey from Gambia to Brooklyn and how his unique upbringing fostered a deep passion for science. We'll also hear about his time at Northeastern, a pivotal internship at Millennium Pharmaceuticals, and his research at MIT under Alice Ting. We'll also chat about his PhD work at the University of Florida, where he studied neurobiology and neurosciences. Without further ado, let's dive into this episode of the Biotech Startups podcast. eMalick, it's great to see you. Thank you for coming on the podcast.
Thanks, John. Great to see you as well.
So we've been really looking forward to this conversation, and I know we've had it on the calendar for a while, but in a kind of traditional, the Biotech Startups podcast kind of journey and opening, we'd love to hear about your early upbringing and how it's influenced your business philosophy and leadership style.
Okay, yeah. So I was raised not in America. I was raised in the Gambia. It's this country that some people know, but most people don't, because it's very small. It's the smallest country in mainland Africa. I live in Brooklyn now, and I love to say that there's more people in Brooklyn than in the Gambia. There's like 1.6 million people in the Gambia right now. Brooklyn is about 2 million.
Those are some crazy stats.
Yeah. And I my early childhood certainly shaped who I am and what I've done. So I was here till 10 years old, then we moved here. But those 10 years were very formational, particularly with my curiosity, what led me to become a scientist. I had parents that were, I guess we call that hippie over here, but over there we just call it unusual. Allowed me to...
Yeah.
They allowed me to ask basically any question that I wanted to when I was a kid, when I was like 4, 5, 6, 7-year-old. There was no boundaries, and they protected me in being able to do that. So oftentimes other adults would tell kids that are asking questions to stop doing that. And my parents would literally tell other adults to not do that around me and my sister. So that gave us a safe space to be inquisitive, to explore curiosity. And I became that annoying kid that was asking about the stars, about the trees, about everything that moves. I had questions about it. And I naturally gravitated towards nature. Was wondering why particular birds went to particular trees during this season or that season, and I was not only asking questions, but I was observing. I was starting to make observations and looking for patterns. So you have to excuse me, I've got a little bit of a cold, so I'll stick.
No worries.
So that led me to in a sense become like a young scientist. That continued on when we moved to the States. I went to undergrad as a major in the natural sciences, like first in biology for a few months and quickly to neuroscience. Neuroscience because biology was wonderful, but when I learned about studying the brain, immediately knew this was sort of like a book that would never have a last page, would be an unending book. So I figured I'll never get bored of this topic.
Yeah, I like this. It's interesting because I always think of it like studying the brain's like the final frontier, but it's like an endless frontier. Like it's just like so infinitely complex. And something about your upbringing like I really love is that, well, there's a couple elements. It's a bummer that adults would try to quell that natural curiosity a child might have. And I've been reading the Rick Rubin, his book on like creativity, and I think he explicitly says is like you need to not lose that, like that childlike curiosity is like something that you need to cherish and nurture, because as you get older, people will continue to like kind of like tamp that down. But that kind of like childlike awe and inquisitiveness and curiosity is what creates, whether it be music, food, science, just like any creative business, any creative endeavor, and most things are creative, is super important. So I love hearing that. And also, growing up in Berkeley, had a hippie, maybe unusual upbringing as well. What you might call unusual. So you're not alone. I feel like a kindred spirit here. So no worries. Yeah, sorry. So I kind of just kind of pulled away for a moment. But you're at Northeastern doing your undergraduate studies. It sounds like you still have this like kind of curiosity, and you'll never be bored studying the brain. Tell me a little bit more about focusing down on that. And did you have a wet lab experience in your undergraduate studies?
Yeah. So the Northeastern experience was great. So first, I would have to give credit to Northeastern. I believe my class was the first neuroscience class in the country.
Oh, yeah. Whoa. That's like that unusual. Like,
yeah, and they didn't tell me this because no one told me.
Yeah, come on over. Just like come on over. We have a wealth of precedent for this.
I'm so surprised that I'm in this very esteemed group. It was a few of us only, but I think 9 of us.
Yeah.
I think those classes are now in the hundreds each year.
Okay.
So yeah, it was just like a really fascinating, experience. It was a combination of biology and psychology. That was what the early neuroscience was, which I guess it is still to this day at its fundamental fundamental core. But it was just, interesting. Every single class that I went to, I was learning something new. And I gravitated towards memory, partly because of me being a child that lived in one place and then moved to another place and having some memories erode that drew my curiosity towards what exactly is memory, and that shapes some things down the line. We could get into that later on. But I was lucky enough to impress one of my professors, and she invited me to work in her lab. And that was my first wet lab experience. Her name was Wendy Smith, Dr. Wendy Smith. I think she's still there as an emeritus professor. So she started this unusual, what's the best way to put this unusual trend in my life. So going into the sciences, going into the world of science, as you progress through the different stages, is a bit like Brownian motion. You're not sure which one, it's like bouncing balls and, but she started this trend where every person that I worked under or was mentored by was either trained by a Nobel laureate directly or had a Nobel Prize themselves.
Oh my God. That's crazy. And just like also you're like, all right, I'm joining this program. Oh, it's brand new and surprise. Like, all-star lineup here.
Yeah, it was, and again, I'm just that curious kid. I did not know any of this. This is all like retrospectively. I was like, oh, and then you realize that, and you're like, oh, then you realize that as well. Yeah. There's a lot of professors that are very, big shoulders and try to take up space. She was the opposite of that. She was instead very given up space. She was very welcoming. I didn't know what a pipette was. And she took my hand and showed me exactly how to use pipettes and how to use precision and how to measure correctly and how to do repetitions and how also not to get hurt in the laboratory.
Yep.
So she was my first introduction into the wet lab, and she was excellent. We've stayed friends since then, and was a key part of my development.
That's awesome. And also, it's like, I always think about, and I love, hearing these stories, because there's like these moments of like these inflection point moments, much like anyone's journey. And it's also serendipitous too. Like you're like, you can only piece together and connect the dots retrospectively, but probably in the moment you're like, this looks cool. like why don't we just keep pulling at this string? And always eternally grateful, and for anyone who's willing to kind of handhold you when you have like, you're just a young kid and you just don't know better, and they're willing to kind of just like handhold you and walk you through it. So that's one, I'm jealous of that experience. That's awesome. And when I went in, it was kind of like 300, the show or the movie. Where
the movie,
yeah. You're like, here's a bit of lions, just figure it out. And I'm like, okay. But awesome experience.
That's amazing, great analogy.
Yeah. You're just like, got kicked off the staircase into baptism of fire.
Yeah.
And so now it sounds like you had a great undergraduate lab experience. And I know you eventually did an internship at industry at Millennium Pharma. How did you like find that opportunity, and how did you secure that first internship?
Yeah, that was also one of the most formative, experiences in general. So I was looking for an internship. Northeastern is known for these co-op programs, which are basically extended internships. And I was looking for one that was the right fit. And we had, thinking of biochemistry laboratory, I had made friends with some of the classmates that were one year above me. And they had interned at Millennium, and I think going back for a second internship. So through that connection, I got an interview, and then I was selected, where I was, again, like, why me? You treated me like, like a pretty like amazing company at the time. They got purchased by Takata, which is a much more mature company. But at that point, they were, I would say, mid-sized company and quite energetic. And this was the first time I was exposed to scientists at scale, where you're walking in the hallway and every person you see is basically a professional in this job. And I was awestruck. It was I'm not sure when people meet George Clooney, this is my version of that. And I tried to play it cool, and I hope I did, but I was awestruck the entire time. And Millennium was a great experience. One, it showed me that science could be fun. So I'll describe that in a second. But it also showed me that I could be successful at science. So Millennium's model was very similar to companies around that time, which is get as large of a compound library of different molecules for the particular targets that you're interested in. So I think at that time they had maybe 100 to 200,000 different molecules, and basically the size of your company's valuation is very correlated to how many of these that you have. And then the next thing is just have scientists go test them out. And so as a junior intern's job was to go ahead and test as many of these molecules for the particular target that we were interested in. And was very lucky to find 2 compounds. I think they were alphanumeric numbers, but the last 3 digits was compound 568, and then 8, and then the second one 806.
That's a wicked memory, by the way. That's like very specific. Wicked memory there.
This was my true first success in science, or doing something where, in a stage where I feel like I'm playing with NBA players, and I gotta get shot, So those compounds did perform like wonderfully, and then it would take it off my hands to go to give it to people who actually knew what they were doing.
Yeah, here we go. Just like, here we go. Yeah.
But
That's
I
amazing.
certainly I did quite well to become I guess the top intern of that period, and it was really encouraging. It showed me that maybe I can succeed in the space. Another thing that was very important there was I got to sit and have lunch together with other people, but with the CEO and talk to him one-to-one.
Wow.
Yeah, it was a really wonderful person. He came from a chemistry background, so he was also went to the academic route and formed a company, which very enlightening for me to know at that time. And he was human.
Yeah. Yeah. Sometimes you just like need that like quick check.
You just
Like,
need
very
that,
tempting.
right?
Yeah.
He's human with a lot more zeros in his bank account than me.
Yeah.
But nonetheless, though, he was human. And when I had a conversation with him, I left the conversation thinking, I could do this too. Which is really wonderful for, I mean, I think I'm 19 or 20 years old at the time, to have that exposure. And he had created an environment, so that point that I said science being fun, he created an environment where we were having fun. Every Friday, like I met him on one of these Fridays where we'd all have these, get-togethers outside there in Cambridge, Mass. And there would be different, foods that you could try. You could play volleyball. There's tons of drinks. I think I was of age at the time.
Yeah.
But in either case, though, people became more relatable, as well as there was some idea exchanges. But also, it was just like you learn about who, their kids will come about and play, as well as like friendships will become deep and in general, just fun. And that itself sort of broke shattered a vision that I had of what scientists, basically what I had was what was portrayed on TV. And it just shattered all of that and actually made it much more welcoming and much more attractive for me to go into the space.
That's incredible. And I think that, and it's like, one, the first thing that I get, obviously, by my reaction is, it's like, yeah, like leadership, like the biggest companies, still people. And I think for me, and now I'm speaking for myself, a lot of like, after having conversation with leaders, so many leaders, it's just like, everybody's just figuring it out. Everyone is dealing with hard stuff. Everyone, and like, everyone's on this journey, and yes, the zeros in the bank account might be different, but still everyone it's the human condition that is like the through line to it all. And that's like, and I at Exeter, we make it a point where we just call them like skip levels, but we always try to like have senior leadership talking to whatever level it may be, and keeping and staying kind of in touch and intimately aware of like what's happening, what's the energy. Because I think as an observer and a kind of history or a historian of business, like when things kind of go awry is when you lose that touch, and you start thinking in spreadsheets, where it's just like, okay, here's how many FTEs we have, or what, and you start managing from a spreadsheet, and that you lose that human element, and then the fun, that's not fun anymore. Like that's like, it's no longer fun.
It's beginning to automate, and you lose people's spirit and their desire to overperform.
Yeah, exactly. And then, as the saying goes, like, I'm not just a number in a spreadsheet, right? So it's like, it comes from that. And I guess for, just for my curiosity, what's like the funness at Millennium, was that like an industry specific thing? Did academia just have a very different for you, your lab experience in academia, just very different for you?
Yeah, I mean, I think it's just the scale. So I've certainly had lots of fun in academia. The scale is different when I was in the industry with Millennium in particular, because I guess there's the funding amounts, and then the amount of budget allocation, and also the competitiveness of. So if you saw what Millennium was testing out on a larger scale with Google a few years later, or I guess until now, to be honest, with the cafeterias, with food, and given giving perks to mothers when they have babies, and having like parties for just literally as like a competitive edge to bring the best talent to their companies compared to the next, right? It's while the universities will try to do things like that, it's a bit more difficult just because the industry of academia is capped much, particularly in the sciences by the NIH. There's only so many grants that you could have. Much of it's going to go back to the university directly as indirect costs and so forth. So what Millennium did and how it was competing is hard for universities. So the universities like, for instance, Columbia versus Yale. Columbia says, we're in New York City, take that, Yale. And Yale's like, we got a house over here, take that, Columbia. So that's that level of competition, right? Versus like, exactly, versus spending $70,000 to make sure your people are happy over a period of a few months every Friday. So it's just like a different space. And I luckily was able to understand that very early on because I saw both sides at the same time.
Absolutely. Yeah. It's like apples and oranges, it sounds like. And yeah, I think I had a similar experience too. I was like, what are these like Friday socials? Like usually in my academic lab, I'm like, maybe we're splitting a pizza. Like maybe. Exactly. You're wrapping up your internship at Millennium. And was that kind of like in your undergraduate study? What year was that? And I know you eventually ended up at MIT. So can you talk a little bit about that period for you?
Yeah. It's so the Millennium was my last internship, I believe. And so when I finished it, because of the conversation with the CEO, instead of continuing to go down industry, which is, what basically all of my peers did.
Yeah.
They were making good paychecks, and I was on par with them. But I decided I wanted to follow this longer vision. Hopefully, one day having something that being a laboratory of my own or a company of my own. So I eventually matriculated to MIT. So how everything looks good in hindsight, but it actually wasn't the easiest. There was this period of time where I was graduating and I didn't have a job, and I had declined taking the interviews at Millennium or any of the other places that I had offered to me just because I wanted to pursue the PhD. So I was going to take this gap year to sort of increase my skill set. And I was in this in-between spot where I thought I was the worst human being in the world. That did happen. My roommates had to help me out with rent for a few months, to be honest. I was like a 21, 22-year-old, something like that, which I thought it was the end of me at the time. So it was bad, but there was one thing that I had in my back pocket, which was there wasn't that many neuroscientists in the world, and I was one of them. Right.
There we go.
So this attracted the attention of this lady, Dr. Alice Ting, who was this young professor at MIT chemistry, and building 66. So I interviewed with her, and she was very young, and her lab was about 6 months old at the time. She had just come from UC Berkeley, to be honest, where you are, in the so here, that's the second Nobel. So she had just come from training with Roger Chen, who was to soon receive a Nobel Prize, and Alice was part of the work that helped significantly with that. He actually named her in his Nobel Prize speech directly. Yeah. And she was just great. We had this conversation in my interview where she said words like awesome and cool and bad. I'm just like, this lady is okay. I could work.
What? Like, this is not what I'm used to.
She just had a really wonderful affect, and she wanted somebody that knew neuroscience, because she had this ambition of using some of the chemistries, particularly click chemistry. That I think now Susan Bortozzi just won the Nobel Prize for that a few years ago. But she was like a very early and forward thinker in using click chemistry to apply labels to neurons so you could visualize in different ways. And this is why she wanted me to come to her lab. So that was a great experience. It was definitely not all positives. It was MIT is a hard place. Like it's it's the high level of give a asterisk like everybody.
Yeah.
So for me, it's really where I saw high performing individuals, and I was sort of thrown into that milieu and like learned to swim, thankfully, and then also became like part of the community, which was really wonderful. And I learned a ton in the laboratory, and made some lifelong relationships since then.
That's amazing. And I want to go back just for a moment about the kind of like things are never always up and to the right on my screen, but I had a similar experience too. And I don't think we're alone in this. Like there are these moments when you're just like trying to figure it out, and in a similar fashion, but in this case, it was my now wife. She kind of, I was trying to figure out Exceeder. And when you have no clients, like when you start the company, you have no clients, like, okay, I can't pay myself a salary. So, and I thought that was okay, I'm starting to just bleed my life savings here. Like, how am I going to do this? And it is the generosity of your friends, family, and loved ones who support you. And I'm eternally grateful for that. Just kind of like was able to smooth that like kind of rough period out. Which, and then, eventually, finally we were able to kind of get on your own feet. But, I had a similar experience too. And in that moment, for anyone who's listening,
yeah,
it's like, you're not alone, right? It's like,
not alone.
You're not alone. Like these kinds of experiences are universal, especially when you are kind of you're going out into that frontier.
Yeah.
There's bound to be these kind of rough patches. So
the waves get stronger, and some of those down waves are really tough. And yeah, if you're listening to this, like it does change. Right? Yeah, but
exactly.
Understand you're not alone. Yeah,
exactly. And this sounds like the MIT lab is like, well, one, it's kind of the smaller nature of the lab almost sounds like a startup within like a larger academic institution, where it's kind of like I'm going to imagine being a small lab, you probably, right, you're doing a lot. You're probably doing a lot, learning a lot. And can you just like talk about what, it sounds like it's a very high caliber, of course, at MIT, but were there any memorable challenges or triumphs for you during that lab experience?
Yeah, there's quite a bit. So I would say the person that I became closest to, just like a heads up. So I came into the lab as about 6 months old. I'm essentially not just coming as like a research technician. I'm coming in to help build the lab up. So like MS machines, like I'm buying and setting up.
You got this. These are also very expensive. Don't screw this up.
Exactly. Hundreds of thousands of dollars. You got this thing out. I'm just like, play it cool. I guess play it cool. And my social life sort of went to zero because I just put everything I can into this. And luckily I didn't screw up. But I became close to Mark Howarth, who was the first postdoc for Alice. So essentially, I got hired, and then Mike got hired. Mark got hired like a few weeks afterwards, something like that. So we were like sort of the early members of the Ting lab, and our benches were side by side, and we became like besties even to this day. Right? He's now the chair of pharmacology at Cambridge in England. So, but he was good at what he's good, So I remember, but there's levels to good. So we were working on this particular assay for something with biotin, avidin, binding to some like a substrate. I can't remember exactly what it was, but a very straightforward protocol that he and I think worked on over, I don't know, a period of time that was significant. And we had this meeting with Alice where we sort of showed up with empty hands. And it could have been just me. Maybe I should drop Mark out of this. It could have been just it could have completely failed.
Sorry,
And I just thought this was like an impossible task. And then the Christmas holidays came about, and went home, and then I came back and we had a meeting with Alice, and then she was presenting all these graphs of how she not just did the essay, but like took it like 100x more than in just a space of like 3 days over Christmas.
Oh my God.
So humbling.
Yeah. I think those moments too, it's like when you see like super excellence, it's like for me, yeah, you're like, oh crap. Like you're talking about the levels, but it's also just like, oh, like it's kind of aspirational too. You're like, okay, like I can push it harder, like be kind of like the foot on the gas, like we can actually do this. And I always think about my co-founder, and when I was still in the lab, and it was a long time ago, but I was a bit kind of like more of like a ruffian like in the lab. But my co-founder kind of like whipped me into shape, and really it was just like, no, like here's what it means to be excellent. And I was like, oh crap, okay, I get it now. It's kind of what you said, being in the NBA. You're like, okay, if I don't pick up the pace here, I'm going to be left behind. It's kind of like a wake up moment too, and you kind of need that sometimes, because like I think before you join the NBA, you're kind of like in the whatever, you're in junior varsity or you're in the G League.
You're like the very best there in the junior varsity, right?
You're just like, I'm just like draining threes in like the G League, dunking on people. You go to the NBA and you're just getting smoked. You're just running on defense every single time. You're just like,
Yeah.
I'm gassed. Like you need to do more lifting and you need to run more in off season.
Exactly. It's like a technique that you need that will change your pacing, that will change your lung capacity, that will change sort of your mentals. But there's like a form that needs to be had for you to achieve that, and like you need that if you're going to be able to maintain the space, right?
Yep. And you know, I can imagine. And by the way, that description of Alice, that like the kind of that sink or swim kind of go hard, that's super Berkeley too. Like super Berkeley. Like Berkeley is not known for hand holding in a lot of ways. So I can imagine, I'm just, you know, I wasn't there, but I can imagine it's kind of like this, it's a high pace sink or swim, like come along, let's go, kind of energy. So that's really fascinating. And so, and also having a mentor like so closely, like kind of being so close on the lab bench is also amazing. Because I think and it's almost like serendipity, right? Like early hires, you just happen to be next to a rock star, and you can kind of like learn via osmosis. So that's very cool too. So as you know, you're now wrapping up your time at MIT. When did you know it was time to, one, I know you ended up at University of Florida, and end up pursuing your graduate studies?
Yeah, absolutely. So I knew, this is a little bit contentious because I'll just be transparent with this. I left the lab and also didn't matriculate to MIT for a very specific reason. I'm an animal lover. Right? When I grew up in the Gambia with dogs and like maybe a cat or two, something like that, and I guess I wasn't okay with sacrificing animals for lab research at the time. I knew I was going to at some point in the future, but I needed to let myself mentally develop to that stage, and I was very self-aware of myself at the time. So I was in the lab for about a year and a half, and remember I was, Alice wanted me in the lab because I'm neuroscience background, but that it wasn't just about advice. It was eventually the experiments were working in silico, or not in silico, in vitro enough that we wanted to start doing it on actual animals. And guess who's the neuroscientist in the room? That would be me. So I was tasked with me and Mark, to be honest, but then it fell a lot on me. So I did the experiments for a bit, but I just wasn't okay with it. And this led to one of maybe the toughest times, again, questioning myself. I had a meeting with her where she's very straightforward, as a person. She's like, how do you expect to become a full neuroscientist, like a Ph.D., if you don't want to sacrifice lab animals? So I think I absorbed that, but stuck to my guns, and sure that I could do it. I did sacrifice a bunch of lives, but then I just wasn't ready for it. So I wrote a resignation letter, which also was called "How Do You Lose Your Recommendation Letter to MIT 101."
Yeah. that is hilarious. Yes.
Right. But I had absolutely no regrets then, and I don't now. It's one of the proudest decisions I've ever done. I knew what I was about. I knew what I hopefully could be able to do, despite losing the support of somebody that was very important to me. Mark was supporting me the entire time, which was incredibly helpful. So that was sort of the ending of a chapter. And also, I really wanted to go study full-on neuroscience, not in the chemistry lab, but in the actual neuroscience lab. And started to learn about, what's it called? Eric Kandel's work at Columbia. Became more interested in that, as well as another professor that was at Johns Hopkins, who was this hot young professor called David Burchelt. And I learned that David was going to be moving to University of Florida. So I started to think maybe University of Florida could be where I'd end up at, because his work was super interesting. Just to continue on with a brownie in motion, he was at Johns Hopkins working in a team that won a Nobel Prize for prions.
Let's go. Let's go.
So protein self-template, and I was he was like trained in the classical diseases in neuroscience, Alzheimer's, Parkinson's, and ALS. And to go back from, earlier I said I was curious about memory when I was an undergrad. I saw this opportunity to go learn more about memory with him at Florida if things worked out. So I had this really difficult period of leaving MIT, just sticking to my principles. And if Alice ever sees this, I always just have to thank her for the wonderful experience that we had together. It was a learning experience for all of us. And also to Mark. Mark was just so wonderful. I was in Cambridge last summer around this time, and we got to have like a really wonderful time seeing the university and having fish and chips and all that good stuff.
Amazing.
But I was destined to be in like a full-on neuroscience laboratory, and just through luck of the draw, it worked out that I ended up at University of Florida in David's lab. I had a co-mentor, Wolfgang Spreit, who was also great. But David was sort of a start of a new chapter where I full-on went into my curiosity in neuroscience.
That's really cool. And look, I and again, I'm speaking for me now. Like, I think you have to you like going against that, like kind of like that principle and that gut feel. I find anytime I kind of like have made that sacrifice, I ultimately regret it, where I'm like, and everyone has different principles. So I'm not trying to espouse anyone has to believe in what I believe in, but when you finally click and you do the work that is aligned with your principles, that's when like magic happens, right? You're like, every day you get out of bed and you're pumped, versus you get out of bed and you're just dreading what you're doing. And, again, everyone has their own principles, and I'm not going to tell anyone to believe in any one thing or the other. That's totally your prerogative. But I completely respect it. And when I was at Berkeley, I did a lot of talks work. A lot of it's in vivo. So, it's hard shit. It really is hard shit. And also, interestingly enough, when I did a little bit of work for Method, the soap company that's at Target. This is back when they were pre-Target. Like they were, they're a startup. Method was a startup at one point in time. And their whole thing, and a lot of like CPG, cosmetics, that whole industry, some of them don't do this, but a lot of them do, is they created like unique in vitro assays to kind of to avoid the need to do in vivo studies. And it got blessed by the FDA and whatnot. So anyways, I'm just saying this, that there are, and especially this was a long time ago, so things have gone way better and more sophisticated. But, you know, I had a similar struggle too, doing a lot of talks work at Berkeley. And so, okay.
Yeah, it's something something folks don't really often talk about within the sciences or biological sciences. But I'm always happy to share my experience, and because I know I'm not alone in it. Of course, grew to do the experiments that were needed to become a PhD, so to speak. Many mice went down. But this is part of a growing process, right?
Exactly. And you're not alone. I had a similar experience too.
That's all for this episode of the Biotech Startups podcast. We hope you enjoyed our discussion with eMalick Njie. If you enjoyed this episode, please subscribe, leave us a review, and share it with your friends. Thanks for listening, and we look forward to having you join us again for part 2 of our conversation with eMalick. The Biotech Startups podcast is produced by Exceeder. Don't want to miss an episode? Search for the Biotech Startups podcast wherever you get your podcasts and click subscribe. Exceeder provides research labs with equipment leases on founder-friendly terms to support paths to exceptional outcomes. To learn more, visit our website www.exceeder.com. On behalf of the team here at Exceeder, thanks for listening. The Biotech Startups podcast provides general insights into the life science sector through the experiences of its guests. The use of information on this podcast or materials linked from the podcast is at the user's own risk. The views expressed by the participants are their own and are not the views of Exceeder or sponsors. No reference to any product, service, or company in the podcast is an endorsement by Exceeder or its guests.